Welcome, everyone. Appreciate you all joining today. I'm going to do a short introduction here and then hand it over to Ellie. Aly Putnam is a postdoctoral research associate at the University of Massachusetts, Boston, and a researcher here at the Stone Living Lab. She is a marine ecologist whose research focuses on invertebrates and seaweed in the context of climate change and climate adaptation. She uses experiments, modeling, and synthesis to understand physiological performance and inform adaptation and restoration strategies in coastal systems. Her additional research expertise centers on biological invasions and their ecological impacts. She earned her BS and MS from Smith College and her PhD in organismic and evolutionary biology from the University of Massachusetts Amherst. Aly, thanks so much for joining us today, and please take it away.
Thank you so much. Appreciate it. All right, I'm gonna need to share my screen. Okay. All right, can folks see my screen okay? All
We
right.
got it.
Excellent. All right. I'm really excited to talk with you all about how we think about, use, and can adapt our field methods to better suit our research needs and be more inclusive and accessible. So the title of this talk is Rethinking Field Methods for Species and for Scientists. So as was just mentioned, I'm Aly. I use she, they pronouns. I'm a marine ecologist, new here with the Stone Living Lab. And I study things like intertidal invertebrates and seaweeds and how climate change and invasive species are reshaping coastal ecosystems. I'm also a disabled scientist. I live with Crohn's disease and multiple sclerosis, both of which are invisible chronic illnesses. They don't always show on the outside, but they definitely shape how I move through the world and definitely how I do field work. And so in this photo here on the left, this is me out in the field installing an experiment out on a rocky shore. And then this photo here on the right is me in an infusion chair receiving my IV immunotherapy treatment just only like 4 or 5 hours after being in the field in that photo. So both of myselves here are part of my science, and that duality, being deeply committed to research and also navigating it with a body that needs care, is what brings me to this talk today. So field work, field science, is really the heartbeat of the natural sciences. It's where so many of us fall in love with ecology and nature. But the way that we design and can schedule it tends to make a lot of assumptions about who can participate. Things like early mornings, long hikes, and physically intense conditions get baked into our research methods without asking whether they're totally necessary, or fully inclusive. This doesn't just affect who shows up, it affects who stays in the field and whose insights are their part of science. And a parallel to this may be a scientific approach that is widely used, but maybe misses some species because they aren't visible using those particular standard methods. So for example, in this photo here of an intertidal site on George's Island in Boston Harbor, our traditional field surveys, where we laid out transects and quadrats, documented little to no Nucella labilis, the dog whelk, a predatory snail, which in 2020 was published to be on a population decline here in the Northeast. So an assumption could be made that Nucella and Boston Harbor are wiped out. Not good. But it turns out they're actually tucked into crevices and other substrates that the field layout with transect lines and quadrats might not have picked up. So these photos here of us working are out on Lovell's Island on a jetty. This jetty is highly structured anthropogenic habitat that we don't typically sample in the way that we would with an open intertidal layout. And here are some example photos of inside the jetty where we see hundreds, if not thousands, of Nucella found with egg cases, which documents that they're breeding population. And we found this to be the case on multiple islands throughout the harbor. So out on Georges, out on Lovels, and particularly there's a huge population of Nucella out on Gallops Island. So if we hadn't gone off of our protocol to look a little closer, we would have reported that Nucella were absent at the site that we had been surveying anyways, when in fact they're quite abundant. And so that's a little bit of a problem. And just like some routinely used methods may miss Nucella hotspots in the harbor, protocols can exclude people too. I've missed research days not because I didn't want to be there, but because the schedule didn't work with my treatment regimen. I've had to skip 5 a.m. starts because my Crohn's disease causes my body to unravel with minimal sleep, or I may miss whole days because after an immunosuppressant infusion, the medication is so harsh, my body just isn't ready to hike through the boulder field that we just looked at on George's. So these aren't just personal inconveniences, they're barriers, structural barriers. So when we talk about designing field methods, we need to ask not just what they help us see in terms of evaluating biodiversity, but who they might leave out. So I'll stay in Boston Harbor for the rest of the science part of our talk, focusing on mixed course habitats belonging to many of the inner harbor islands. These photos here come from Lovels Island and is a beautiful example of the habitat that I'm going to be talking about. So just very briefly, I want to mention that this work took place across many Boston Harbor islands, and it was a collaboration between USGS, the Northeast Climate Adaptation Science Center, which is housed in UMass Amherst, and the National Park Service from 2020 to 2024. So we know that the harbor is a really dynamic system with a mix of low wave and high wave energy areas, plenty of urban influence, and a lot of ongoing management and even restoration. And of course, Boston Harbor has a pretty well-known history from being heavily degraded to now being considered one of the big success stories in coastal restoration. But at the same time, these systems are now facing a new set of pressures, especially with climate change, sea level rise, and increasing disturbance. The Harbor Islands were listed as one of America's most endangered historic places due to climate change, leaving these erosion-prone islands in jeopardy. So the Boston Harbor Islands are part of a very cool glacial legacy. That is what developed these islands. These drumlin islands were formed by glaciers over 20,000 years ago. So they were shaped by ice, gravity, and time. Most of them are made of layered sediments that were deposited and then molded by glacial retreat. So most of them are not solid bedrock. And a fun fact, the Boston Harbor Islands archipelago is the only glacial drumlin archipelago in North America and is only one of 3 globally. So because of this glaciation, Boston Harbor spans a range of intertidal habitats from marshes like we see on Kathleen Stone Island to permanent bedrock like the outer islands, Calf, Green, and Outer Brewster. But what dominates many of the inner islands is this middle ground, this mixed course substrate. And this is the habitat type that I am going to focus on. Its mixed course substrates differ in structure and maybe even biodiversity, and so requires tailored approaches. Now, when we think about intertidal systems, we often picture something like this, a pretty structured, solid bedrock environment with clear zonation patterns, where the upper intertidal is dominated by barnacles and snails like periwinkles. Our mid-intertidal is usually brown seaweeds like Fucus and Ascophyllum, and then red seaweed and kelps are in the lower and subtidal region. And this is what many of us picture when we are thinking about intertidal fieldwork. Here we are in Gloucester, Massachusetts, actually, using a standard field method that is really built around this kind of system where the zones are visually distinct, like here we are sampling in the mid-intertidal fucus zone. And then in this photo on the right with the quadrat is placed on top of a seaweed community, which is sitting flat on flat rock. So it's relatively easy to sample. But with mixed course habitats that have structural complexity and dimension, this might not translate as well. So here's an example of what a mixed course intertidal actually looks like. This is a site on George's Island. Instead of continuous bedrock, we've got this really heterogeneous mix with cobbles, boulders, sand, some broken seawall, everything kind of overlapping with each other. And even though we do still see some biological patterning, it's much more patchy and discontinuous compared to those classical zonation systems. We can see some banding of seaweed, like we see this ascophyllum zone here and a fucus zone down here, but the structure is patchier and in different areas of the intertidal, right? So, for example, ascophyllum is not found in the mid-intertidal here with the fucus, as we saw in some of our previous photos. It's actually up here, kind of in this upper intertidal area. So we're not seeing classic zonation patterns the way that we see in solid bedrock habitat. So instead of seeing clear delineation in zones, we're dealing with a mosaic essentially. And here we are back out on levels. See how beautiful this mixed course intertidal is. And when we zoom in and look closer at the substrate, we have a three-dimensional substrate. So unlike that photo from Gloucester where the biodiversity is sitting on top of a solid rock slab, here the biodiversity is on top, it's around, and it's under all of the different layers of sediment that we have here, rocks, boulders, gravel, etc. So given this mismatch between habitat and traditional expectations, we started thinking about how existing methods actually perform in this mixed course substrate. So there are already some really strong, well-established protocol for intertidal systems, both on the West Coast and here in the Northeast. And so our project reviewed a range of these approaches and selected three in particular that represent different sampling strategies to then test out in this mixed course habitat. This includes the National Park Service Northeast Temperate Network Protocol, the Northeastern Coastal Stations Alliance, or NEXA approach, and the Stone Living Labs protocol. Each of these is designed with different strengths and goals in mind. So it wasn't about picking which one is best, but looking at how they all perform under the specific conditions. And what we were really interested in was, what does each protocol capture well? And what that variation might be like in our patchy system, like mixed course in the Boston Harbor Islands. And is there a way to combine the strengths across approaches? So we took a test and adapt approach rather than starting from scratch. So the NEXA protocol, it uses fixed quadrats that are placed at consistent intervals. This does give really strong repeatability over time and works well for tracking change at specific locations. But in a system like mixed course, where patchiness happens at small and large spatial scales, and sometimes the substrate shifts and it moves, that fixed placement may not always capture the full variability. The National Park Service protocol is organized around biological zones. So sampling is structured based on where these organisms are expected to occur within biological zones. That works really well in systems with clear vertical zonation, but as we saw earlier, those zones are a little less discrete in mixed course habitats. And then the Stone Living Lab protocol takes a broader spatial approach with much more flexible placement of quadrats across the site. That allows it to capture more of the heterogeneity that we see in the systems, so it's particularly well suited for capturing variability across the landscape. It spans a 48-meter length of a field site, which is pretty far, but is important when thinking about that heterogeneity of the field site. And so I want to mention that there are multiple components to the three protocol that I just briefly described to you, but I'm just kind of talking about these three at a very high level perspective for the sake of time. So what this figure here is showing you is how we laid out our sampling across a shoreline. So this is a field site in Excel. If you can think about the Boston Harbor and put it in an Excel spreadsheet, this is what it looks like. Each of these small squares represents a potential quadrat sampling location, and then the colors represent different biological zones. So we've got our ascophyllum zone, our mussel zone, our fucus zone, and our red algae zone. And so this is the field site out on George's Island that I showed you a couple moments ago. And then on top of that are the numbered points that show where our sampling quadrats were placed and to which protocol they belong to. So you see these yellow quadrats here are the sampling for the Nexa protocol. The purple quadrats are associated with the Stone Living Lab protocol, and then the pink outlined quadrats are those that are associated with the National Park Service protocol. So essentially, we're layering multiple sampling approaches onto the same site to compare how they perform. We tested the 3 different methods across sites on George's Island and then used multivariate analyses to evaluate which parts of each protocol best captured biodiversity in this system, and then took those components and built our new protocol. And so without showing you all the math gobbledygook, I will show you what the final components of our protocol that came from the other 3 protocol ended up being. So based on our statistical results, we developed a survey that retains the strengths of those 3 protocols and captures the variability within our system. So we used a 48-meter horizontal transect and divided it into a 9-quadrant grid, much like the Stone Living Labs protocol, to maintain consistent and broad spatial coverage. And then within that grid, quadrats were randomly placed to avoid sampling bias. And importantly, we found that incorporating biological zones, like how the National Park Service protocol prioritizes, was actually really critical. So at each site, the zones were first delineated, and then we added additional quadrats into these zones. So we have essentially in this field site, we had 36 sampling quadrats to evaluate the biodiversity at this site. And so looking back at the field site at George's, here is our protocol laid out for data collection. You can see that our transect is laid out 48 meters. It goes from this right side of the photo all the way almost out to this outcropping of rocks. Really far spatial coverage. And then within this, we have our quadrats that are distributed throughout the grid, which we marked with these little hats, these little yellow hats. So you can see we've got yellow hats in the ascophyllum, and so we will bring our square grid to sample at these hats in particular. So this setup lets us systematically cover the site while still capturing the patchiness that we talked about earlier. All right, so the good stuff. Using our adapted methods, we were able to capture a broad representation of the intertidal community. So here in this graph, I'm showing you the number of species that have been detected on George's Island since as early as 2009. And in 2022, when we sampled using the different protocols that I was just describing to you. So we're seeing a tenfold increase in species detection in 2022 compared to the other years. And it's not that these intertidal species just showed up in 2022. They were likely always there, but this reflects our increased spatial coverage and our targeted sampling efforts, which gave us the ability to capture more of the fine scale variability within our sites. And so in 2023, we used our new protocol to collect biodiversity data at 5 islands of importance to the National Park Service and sites that we collaboratively identified. We evaluated sites that we determined to have high biodiversity and those sites of cultural importance to National Park Service, usually coinciding with some level of shoreline erosion where action may need to take place in the future. So for example, this erosional area on the northeast end of Peddix and on George's Island, where we see the corner of Fort Warren sitting dangerously close to erosion that has impacted the seawall. And so similar to the results that I just showed you for George's Island, most of our other project islands that we surveyed, we more than doubled the number of species detected from historical sampling on these islands. So Gallup's Island, though, was our most biodiverse island, with George's not being too far behind, and relatively high biodiversity, across the islands like Peddix, Levels, and Thompson. And just to ground you in what we actually saw, here are a few examples of species that we documented in these surveys. So here I'm showing you a mix of encrusting bryozoans and colonial tunicates, which are often in really patchy distributions and under the substrate side of rocks. We had a variety of gastropods, such as this tortoiseshell limpet, which is actually thought to be in decline across the Northeast region, but we were very happy to see that it was quite abundant at our sampling locations and in other areas of Boston Harbor. We found a variety of echinoderms like brittle star and our sea stars. We also saw a range of crab species, from natives like the rock crab and spider crab to invasive green crabs. And across macroalgae, we're capturing both native and introduced species, often co-occurring within these patchy habitats. And so beyond visual surveys, we incorporated other methods into our toolkit to better understand the community. So sampling other methods like crab surveys, photoquadrats, and eDNA. Each of these methods add a new layer of information, helping us to better understand species presence, abundance, and community composition. So first, starting with our crabs, we used multiple methods to catch crabs because no single approach captures the full picture. Different species use the intertidal in different ways and at different times. Some are more likely to be caught by hand under rocks, while others are best found in traps or caught overnight. So if we only used one method, we miss key species or underestimate their abundance. And so that's why building a diverse toolkit is essential to monitoring in general. So for example, we used band transect surveys to track species like the invasive Asian shore crab, Hemigrapsis sanguineus. Here we cover broad spatial area and target specific habitat nuances. So we travel along this transect line 1 meter on each side, and we're flipping over rocks and seaweed and hand collecting individuals that were tucked away underneath. And as you can see, when we compare the number of Hemigrapsis caught via the band survey versus traps, we caught a significant amount of crabs with that hand method versus a trap. Other crabs use the intertidal but are not caught as often via band transect. And so another method that we deployed was trapping. We placed traps baited with squid along our vertical transect lines in the high, mid, and low intertidal. These traps set out for a full cycle, a full tidal cycle. So they were underwater during high tide and then exposed at low tide, and then collected the following day. And these baited traps were especially useful in collecting invasive green crab like Carcinus manus. Compared to band surveys, they were significantly caught more in traps versus by hand collection. So this highlights how different methods capture different parts of the crab community. Other species that we caught in our traps were the rock and Jonah crabs. So to monitor abundance, or more accurately for certain species, we need to use multiple or different approaches. We also use photoquadrats to capture high-resolution images of the benthic community. This lets us analyze percent cover, species ID, and community structure back in the laboratory, which offers a non-extractive way to track changes over time. So essentially, at the places that we manually calculated biodiversity, we also snapped a picture nearby to look at later at a different time in the laboratory. And so here's an example of those really close-up photo quadrant photos. We can see that we've got mussels, we've got tunicates, we've got seaweed, we've got limpets, all within this really small area. And so this is a useful way to look at the intertidal while not having to physically be there. Could be used by community scientists, could be used as a training method. There's a lot that can be used for by photoquads beyond what we did with this project. And so environmental DNA, or eDNA, is another tool we added to our biodiversity monitoring. It refers to DNA that is shed by organisms into the environment, like through skin cells or waste, and can be found in places like the intertidal zone. And so at each of our sites, we collected water and sediment samples and then extracted, amplified, and sequenced the DNA to identify which species were present. Our goal was to see how well this method can detect intertidal biodiversity compared to our other methods. And so interestingly, eDNA picked up the greatest number of species overall, including many vertebrates, likely using the intertidal zone during high tide, like all of these different fish species that I have in this photo. While there was pretty limited overlap with the number of species that we found in our visual sampling versus what was actually found in the eDNA, that's okay. This tool is not to be a replacement, but actually a complementary tool to understand the whole ecosystem, because things like fish and other species that use the intertidal, when we're not out sampling, we were able to capture that. So by adapting our methods to match the habitat and including multiple other tools, we are getting a more complete and holistic picture of these communities. And so far, I've been talking about adapting our methods to better match the habitat, but there's another layer to this. Thinking about who those methods work for and who gets included in this type of work, because field science isn't and shouldn't just be about the data. It's also about who gets to participate in creating it. And so these photos here represent two accessibility efforts that I wanted to highlight. On the top, we here have a boat. An organization bought this boat that was physically redesigned to welcome wheelchair users on coastal expeditions. And on this bottom photo here is a team that helped create American Sign Language signs for marine terms that had never existed before. The term, believe it or not, the term estuary did not exist in ASL until 2018. So inclusion in fieldwork means adapting tools, language, and spaces so that everyone can take part. But I want to briefly reframe how we think about access in fieldwork. Disability is incredibly diverse, including physical, sensory, cognitive, and learning differences, some visible, some not, and often intersecting with other identities like race, gender, and class. And importantly, it's not just about individuals, but about the barriers that exist. In fieldwork, those barriers can come from the environment, the equipment, or how we structure our work. And this becomes very real in the kinds of environments that we work in. So for example, this is one of my research sites in Narragansett, Rhode Island, and many rocky shores sites like this are uneven, unstable, and physically demanding to navigate. So even just getting to a sampling area can be a barrier. So going back to this idea that people are not solely limited by their bodies, but by barriers, in fieldwork, those barriers can look like this. They can come from physical environment, from logistics and resources, and from cultural and expectations around fieldwork. And in fieldwork, we can start to break those down a bit more specifically. And these barriers show up in a few key ways. There are physical and logistical barriers like accessing field sites, dealing with terrain and weather, or having limited access to adaptive equipment. There are also health and sensory factors like fatigue, chronic illness, heat, or sensory overload in complex environments. And then there are social and structural barriers like assumptions about ability, stigma, or inflexible expectations in how fieldwork is done. But most importantly, many of these are not fixed barriers. They're things that we can design around. Some of the simplest ways to expand access are through tools, but also through how we collect data. For example, using this photo-based quadrat that I described earlier in the talk allows for data collection without requiring constant bending, fine motor work, or time pressure in the field. There are also tools that support communication and data collection, like tablets or voice-based tools or visual identifiers, which allow people to interact with the environment in different ways, in ways that suit them. Mobility and support tools like seating or transportation equipment can make a big difference in how long and how comfortably someone can work in the field. And things like noise reduction or other sensory supports can help make field environments much more manageable. A friend of mine with some sensory challenges has described how these noise canceling headphones made her fieldwork out on a variety of islands that have thousands of screaming terns, the shorebirds, just made it so much more manageable because she could cancel out these squawking birds and focus on her work. So these aren't just add-ons. There are ways of adapting field methods to expand who can participate. And accessibility isn't just about equipment. It's also about how we choose and set up our field sites. A big part of accessibility is just planning early rather than waiting until someone has to ask for accommodations. So that's where taking time to learn about accessibility and how you can adapt your work is really important because you can be proactive rather than reactive. Things like stable access, space to rest, or even basic needs like hygiene, access to restrooms, and medical support can make a big difference in who's able to participate. And in some cases, that means choosing sites or modifying research goals to make physical visits more accessible. For example, choosing sites with stable access or infrastructure like ramps, as seen in this top photo, or maybe choosing to add on a project that happens in an environment where sampling can happen in a more controlled and accessible way, as seen in this bottom photo where dock fouling research is happening. And when you do that type of research on a dock, that usually includes ample parking, easy access to the location, and is less dependent on tidal cycles, so work can happen at accessible hours. All of these decisions, these tools, these methods, and site choices really do shape who is able to participate in field science. And so why does inclusive fieldwork matter? Well, first, I think it's important to make fieldwork inclusive because at the core, it's just the right thing to do, right? But beyond that basic principle, which is, I think, the most important at its core, is that when more people can participate, we're not limiting who gets to contribute to research. And so then different perspectives that are brought to the table can change how we design our studies, what questions we ask, how we interpret our data. Um, Even in our work, the Boston Harbor Project I was just describing to you, adapting methods like using photo quadrats wasn't just more accessible, it certainly was, but it also gave us more flexibility in how we approached data collection. And to be honest, when we had sick or injured interns who wanted to still contribute, this allowed for their inclusion and the continuation of data collection. So ultimately, by being inclusive and bringing more people to the table, it makes for better science. So I want to end with just a few things to keep in mind, especially for those of us working in field science or supporting field research. First, it starts with your mindset. Respecting all disability, including what isn't visible, is really important, as well as not making assumptions about what people can or cannot do. Um, Making sure that disabled scientists, students, participants are actually included in decisions and not just accommodated for after the fact is really important for creating an inclusive space. And then in practice, supporting inclusive fieldwork, highlighting disabled scientists, and being responsive when access needs comes up is really important. Accessible fieldwork isn't separated and shouldn't be separate from good science. It's part of it. And so with that, I would like to thank you all for letting me talk to you about these two issues. And we're going to put a link in the chat to some resources. I've got a variety of resources, reading resources for folks to learn more about accessibility, disability in fieldwork. And then I would like to plug, it's at the very bottom of the document. I'd like to plug that there's a new book that will be coming out called Inclusive Nature, which is about making nature open and accessible to all. And in one of the chapters, myself and a couple of my colleagues wrote a chapter on disability and accessibility. So that's currently out in review by Johns Hopkins Press, but will hopefully be out in the world soon. So, but there's a link there for you to read more about it. And with that, I will say thanks and take any questions.
Thank you so much, Aly. That was absolutely incredible. Really appreciate the presentation and all the insights shared. Would love to have some questions from attendees. So if folks have a question, please just raise your hand and we'll go through them that way. And who wants to kick us off? I see Alana's hand raised. Yeah, please.
Hi. How revolutionary do you feel like it is to talk about inclusive fieldwork methods? In archaeology, it's pretty revolutionary, and I
Oh.
wonder if you've ever faced pushback about it.
Oh, that's a great question. And Alana, I'm so happy to see you on Zoom right now. Alana and I have worked together in the past when she was an undergraduate and while I was doing my PhD. So really glad to have you here. That is a really great question. It is not something that is talked broadly about. And if we just think and talk about the history of disability and accessibility, it has been very stigmatized. So um, I talk about it as often as I can, where I can, because it's important to try to change the field to be more inclusive. I've had a lot of really great reception to the things that I've brought to the table around making changes for accessible fieldwork, and then sometimes less. And so we just don't give up the fight. So.
Thanks so much, Ali, and great question, Ilana. We have a note in the chat from Mark Albert, who says that he's on public transit right now, so can't be on audio or video, but said, wonderful talk, Ali. The combination of content was powerful. And he asked a question as well. Might the lessons from the protocol assessment bear on the Stone Living Lab's living seawall designs for use in Boston Harbor?
That's really, so if I'm understanding your question, hopefully the way I respond means that I understood your question. The living seawall, I think, is exactly what I'm talking about in needing to adapt our methods and how we do things um, to meet changes that are happening, right? So this is a nature-based solution that is very creative in its design, that was designed specifically for the goal in mind, right? And that's the idea here that I've been talking about throughout this presentation is needing to make changes specifically to meet goals and be inclusive. So I think that the Stone Living Lab's seawall designs is a perfect parallel to what we're talking about. And thanks for being here, Mark.
Thanks, Aly. Other questions?
Yeah, I'm curious about the time that's needed to adopt your protocol, which I like. But do you find it takes more time to apply your protocol compared to some of the other stuff that is out there?
You mean the actual like field
Yeah,
protocol.
yeah,
So our protocol does have a little bit more, it does have several more sampling quadrats than some other protocols that are out in the world for our region. So it does take, I would say it does take a little bit more time, but it's the trade-off I think is worth it because we had this broad spatial coverage. And as I described about the patchiness of the mixed course intertidal, we do need to cover broad spatial area. Otherwise, we miss things that are happening at fine scale due to the nature of the substrate. So yes, it's a little more time intensive, but it's a positive trade-off. But depending on what your goals were, right? So like our goal for this project was um, to develop a baseline of biodiversity at these sites. So it was actually really important for us to make sure we were thorough and could capture as much biodiversity as we could so that when we handed that list over to National Park Service and USGS, that they had a full picture of what the biodiversity was like so that when they make decisions on what type of adaptation efforts they might need to enact or if they need to physically do anything at these sites, they have an understanding of the community and can have pre-data and then be able to look at post changes.
yeah, yeah. No, I love the approach. Um, in my career, I've been asked a number of times to step in and review field research, and I always start with, so what are your biases with this method?
Mhm.
And usually they say, I don't have any. Well, yeah, well, let's talk.
Yeah.
All right. Because every choice, I mean, you've come pretty close to eliminating the biases, but a lot of the time, especially as you get back in the day, since we're different generations, the protocols were pretty simple, but not always very accurate. They were a lot of times convenient
Mhm.
for people,
Yeah.
right? And we missed a lot of information and came to a lot of conclusions that weren't very helpful.
Yeah.
So thank you. Great job.
Yeah. Thank you. That's a great question. Appreciate it. Thanks for being here.
Thanks, Don. Thanks, Aly. Um, Additional questions? Katie.
I just, reflecting on how important baseline surveys are for understanding the consequences of stressors or beneficial interventions. I just wondered, in terms of developing your protocol, and how did you think about the spatial scales of when we monitor to make sure that we're always capturing the right information, and whether there had been kind of discussions around responsibilities for both ownership of those sorts of data sets long term and continued implementation of the protocols long term?
Yeah, that's a great question. So starting with the spatial scale component, I think we can't take a transect and sample the whole island, right? Like, I have walked the perimeter of Pedix, and that is exhausting. So to sample the whole island is not realistic, right? But having broad spatial coverage, some of the other protocol that we had assessed had much smaller spatial coverage. And that was a very key outcome in our statistical analyses, was that the smaller the field site, the less species diversity and richness we were getting. And it was, inspired by the Stone Living Labs protocol that you all use out on Rainsford and whatnot, to have a broader spatial coverage of 48 plus meters. And that was a key component to this work. And then it's the conversations of, who we're working on projects with and where sampling should happen because of, erosion happening at a particular location where action may need to be taken, right? Like having a conversation with practitioners and stakeholders. We did not pick our field sites just because we thought they were scientifically interesting, for example. We sat and had discussions with our partners from National Park Service, hey Mark, and discussed what was important to the preservation of these islands, and also that biodiversity is part of the islands. It's not just the structural, anthropogenic components of the islands that are important as well. And so then the second part of your question, I think, was about data and who has what data and repeatability. So given that this project was a USGS National Park Service project, all of our data is available publicly through ScienceBase, which is the USGS repository. So feel free to go check out our species list if you'd like, do some fun things with the data as you wish. And I think that having a place, what I've appreciated, things in federal science are very complicated right now. But what I've appreciated in my experience of working as a federal employee on this project is the data availability, the data accessibility, that everything is out and it's in one singular place. And I think that's important, rather than having data in someone's drawer that never got out into the world, or in someone's GitHub repository that they forgot the password to, and then now they can't get into it. And so I know and appreciate the data aggregation for the Stone Living Lab anyways, and making it accessible through your website around tidal height and weather and whatnot. So and then the repeatability, it's really challenging to be in federal science right now with our current administration, and funding is a real challenge. And it does cost money to pay people to be out, to take a boat out to the islands. We use actually fairly less expensive equipment than other types of science, like a 5-gallon bucket from Home Depot versus like some genetic lab equipment. But it is costly, and some of our methods, like eDNA, for example, have a little bit of a price tag to them. And so finding funding to be able to repeat that is a challenge. And that's not just our challenge, it's a challenge across marine systems and coastlines. So sometimes I'm always happy to go out and sample for free, but I'm only one person and we need, like 10 people on our team.
Thanks, Katie. Thanks, Aly. Um, any other questions from the audience? Free to raise your hand, jump in. Katie,
One
have at
more.
it again, please.
It's because you mentioned eDNA, and I know you, like me, are a bit more of a traditional morphological taxonomist. And so now that I hear you've had these experiences with both methodologies, and there's always that contrast between cost and time taken, and what information do we gain from each approach? Just wondered if you could comment on that in respect to your protocols a little bit more.
Yeah, absolutely. The eDNA part of the project was really fascinating because eDNA is this very hot, fun, exciting new world. And it certainly is, don't get me wrong. But it's a little more complicated in marine systems depending on what question that you're asking. So we wanted to know, could eDNA help us understand the big picture of intertidal diversity? And, basically, does it match what we were seeing in our visual quadrats that I myself, as a intertidal ecologist, community taxonomist, looking at identifying and writing on paper, can we use eDNA samples to streamline the process? Does it match one for one? And it performed pretty poorly, if I'm being honest, in terms of intertidal invertebrates and seaweed. My graph, I think it showed that we had only 14 species total of the 60 that were identified at a particular location. Only 14 of them were found by the eDNA sampling method. But the eDNA sampling method gave us a whole set of information on other organisms that use the intertidal that are not in our quadrats. So we had a whole beautiful set of data on fish and other vertebrates that inhabit the area. We documented sturgeon in the harbor based on that. And so really, in this case, we say, no, it cannot be used as a replacement for traditional intertidal monitoring and surveying, but it can be used in tandem, in conjunction, to understand like the whole ecological picture, who else is here in the intertidal.
Super interesting. I found tiger DNA once in a sample from Sydney Harbour, but then realized I was right next to the zoo, so it probably
Yes.
washed in.
Yeah, yes, exactly. I think we did have one set of results come back with chinchilla. And we were like, is there a chinchilla population on the Harbor Islands that we didn't know about? But I think it turned out that there was some cross-contamination in some respect. So no chinchillas in the intertidal. We've seen deer in the intertidal in the Harbor Islands, but no chinchillas.
That's great. Let us know if you see any tigers in Boston Harbor as well.
Will do.
Cool. We got a couple minutes left. If there's any additional questions, great, but okay to break a few minutes early as well. I see a few folks signing off. Any final questions before we break? Well, thank you, attendees. Thank you again, Aly. This was fantastic. Really
Thanks.
appreciate it. Appreciate you. And everyone have a great rest of your afternoon.